So in this video, I'm going to go over how to break down experimental designs and how to make flowcharts while looking at these designs. I want to start with a disclaimer that I will be going into much more detail than you would actually need to go on the MCAT. And I will break down each figure that comes up so we have a knowledge on how to break down figures and how to derive meaning from the data that is being presented in these figures. We have our passage down here, and we have multiple figures. We have 4 of them. I will break down each of them, and then further down we have our questions. And we want to do a breakdown that is efficient enough where we do not need to keep scrolling back to the passage, but we can just look at the breakdown. So that's why it's very important for you to do an accurate breakdown, because if you derive the wrong meaning from it, then you'll be answering the questions wrong as well. Let's get started. There are selective neuropeptide uptake modulators that are drugs that can alleviate symptoms of depression by blocking the reuptake of the neurotransmitter ZNT, thereby increasing the amount of the time ZNT remains active. Elevated levels of ZNT within the synapse are associated with feelings of well-being. Conversely, low levels are correlated with depressive symptoms. Recent studies have shown that SNUMs can also mediate their antidepressant effects by increasing brain levels of certain cytokines, including modulin beta. So now we have multiple factors going on, so we'll start with our breakdown over here in our gridded paper. On the actual MCAT, you will also receive gridded paper, so this is where I would write what I would actually write on my sheet during the actual MCAT. ZNT reuptake blocked by SNUMs, which also increase MB, which is our modulin beta, and that is our cytokine. Further on, it says MB directly increases the expression of the adapter protein PX7 in neighboring neurons, which then interacts with ZNR5, a transmembrane receptor for ZNT located at the neuronal surface. All right, so we'll finish up our flow chart. So there, MB increases PX7, a right protein, which directly affects our usually I write, this is my transmembrane protein receptor picture. So this is ZNR5, and then ZNT will interact with that. So this is my basic chart. Anything else that I learn from the experiment, I will add on to this flow chart. Let's go to figure 1. Figure 1 is showing ZNR5 expression in the membrane-enriched hippocampal fraction and the total lysate from wild type and PX7 knockout mice. So this is looking at the relationship of, so our independent variables, this is also helpful breaking down. So independent variables are knockout versus wild type, and our knockout is no PX7 and then plus PX7. Our dependent variable is levels of ZNR5 in membrane versus total. I'm not writing this down on my gridded paper, but it's something that I'm making a mental note of. So figure 1, when we evaluate it, the total membrane levels are different. They're significantly different. The star means star equals significant difference. But the total lysate has no difference in how much ZNR5 expression there are, but on the membrane, there is a significant difference when PX7 is knocked out. So figure 1 is telling us that PX7 leads to localization of ZNR5. No change in production. The amount produced is still the same, but the localization on the membrane is different when PX7 is knocked out for ZNR5. Okay, moving on to figure 2. cAMP levels in cells transfected with ZNR5, our membrane receptor, and or PX7, so in conjugation with PX7 or separately. So we can again do our independent variables, which you can always pause the video at any point and try to do it yourself. So our independent variable is ZNR5 plus PX7 or PX7 just ZNR5. And then the dependent variable is cAMP levels. I also want to look at the trigger words when they're showing up. cAMP is a trigger word for G-coupled protein receptors. So G protein coupled receptors, GP. Let me rewrite that. GPCRs. This is just something, again, I'm making mental notes of, but on our paper, I'll write figure 2. Figure 1 over here. Figure 2 is showing us that there is a significant difference when ZNR5 is alone administered versus in conjugation. There is a significant increase when they're in conjugation with PX7. It's pretty close to control. So we know that PX7 enhances ZNR5 signaling. So there's some codependence happening, and it's probably because PX7 is helping it localize. We'll move on. Studies have shown that certain analgesics, such as Novaprax, can interfere with the behavioral improvements typically observed in mice receiving long-term SNUM therapy. And then figure 3 shows effects of Zepralin, which is a known SNUM, and NPX, which is supposed to interfere. All right, now that we have our flowchart again, we can look at figure 3 and what it's trying to tell us. Our independent variable in here is our ZEP, which is SNUM, then NPX, which is our inhibitor. So I'll write a little inhibitor sign, and then both of them together. So can one overcome the other? And our dependent variable is changes in MB. So we are looking at our flowchart. We know if one changes, the other one will change. And whatever the results of this is will also confirm that our flowchart is correct. So we are seeing that when ZEP is administered by itself, the fold of MB is much higher than when NPX is administered. And then it's still pretty low when both of them are administered. So ZEP is not able to overcome the effects of NPX, is what it's showing. So in our flowchart, we'll also add this. NPX is doing an inhibitory effect to MB. So figure 3 shows that NPX interferes with ZEP's ability to increase MB levels. ZEP's ability to increase MB. Okay. Now figure 4. Figure 4 says PX7 expression in vehicle and Novaprax. So is NPX affecting PX7 expression as well? We know it's affecting MB expression, and then is it doing downstream to PX7 as well? Or is there some independent pathway that PX7 is increasing by? Okay, so our independent variables here are vehicle, which is no treatment, so it can be our control, and then NPX, which is our inhibitory. Our dependent variables is changes in PX7 expression. When you're looking at figures, we really just want to look at where there is the significance and like where's the significant difference. So in NPX, there is no significant difference between the vehicle and the ZEP administration, but between the vehicle and the NPX treatment, there is a significant difference when ZEP is administered. So this reaffirms that NPX is lowering PX7 expression by targeting the same pathway that ZEP is also targeting. So this is saying that NPX is in the same pathway as ZEP. Okay, so figure 4 is saying that figure 4 NPX suppresses. It's suppressing ZEP's effect on PX7 expression. And that's probably by lowering the levels of MB. But that's just something I'm assuming. Okay, we'll move on to the questions, and I'll move our flowcharts and our figure evaluations down to the questions so we do not have to keep going back and forth to the passage. But if there is something that we really need from the passage, we'll go back up. Okay, moving on to the questions. Based on the information provided, ZNR5 most likely belongs to which class of membrane proteins? Before, when we were breaking down each figure, I pointed out that CAMP levels were being measured, and that is a keyword for GPCRs. I've added them to our breakdown here, and we see that GPCRs is one of the answer choices, so we will pick that and move on. Protein kinases usually will have keywords like phosphorylation of some sort. We didn't talk about that at all, so we will ignore that. Tyrosine kinase receptors we'll talk about. Again, phosphorylation, tyrosine, even the hydroxyl group, but that wasn't talked about. Ligand-gated ion channels we are not talking about ligand-gated ion channels because there is no change in potential or any type of ion being measured. So D is our best choice, and we'll move on. ZNT is synthesized from a single amino acid by a short metabolic pathway consisting of a hydroxylation reaction followed by a decarboxylation. And the chemical structure of ZNT is shown. From which amino acid is ZNT synthesized? For this, the best way to do it is what does this look like the most? Which amino acid does it look like the most? And we know it's tryptophan because on the MCAT, we need to know our amino acids like the back of our hands. So proline will be folded upon itself. It will have one ring. Tyrosine is like phenylalanine with an OH. Histidine is also, again, one ring. It's the imidazole ring. And this is an indole ring, which is present in tryptophan. So we'll pick that and move on. In an experiment to determine whether NPX alone causes depressive-like symptoms in normal mice, which of the following would be most appropriate control group? These questions will show up pretty often on the MCAT, so you need to know how to do experimental designs. What are they trying to find out? So if NPX alone, our study is NPX alone, depression, does it cause depression? This is what we're trying to find out. Our independent variable and our dependent variable. Our dependent variable will be symptoms of depression. Are there symptoms of depression? And then for our independent variable, we'll need two groups to compare. One will be the control, and one will be the administered treatment. So it says in normal mice up here. So our independent variable would be normal mice plus NPX. So that will be compared to a negative control, one that we will know is not going to produce the results. So it'll be normal mice with a placebo, a vehicle of some sort, right? Something that's a sham treatment, just like NPX. So that will be normal mice treated with vehicle. Now, we can cross out many of these. Depressed mice, we want to know if depressive-like behavior is present in normal mice. So if we're comparing it to depressed mice, we are not able to make an accurate decision. So anything that says depressed, we'll take away. And then it's asking if NPX alone will cause depressive symptoms. So if we're comparing it to ZEP, that's not allowing an accurate comparison. So that will give us confounding variables, so we do not want that. So C is our best answer. Okay, moving on to question 4. Which comparison would best determine whether MB is necessary for the ZEP-induced increase in PX7 expression? So we know that when ZEP is administered, that will increase MB, and that will increase PX7. So back to our flow chart, ZEP is an SNUM, so you can write ZEP here. And then, is MB even required, or can it just directly increase PX7? So our requirements for experimental design, this is our experimental design. We want an independent variable that has MB, and then it doesn't have MB. And then our dependent variable would be changes in PX7, and another addition is plus ZEP treatment, right? You need ZEP to be treated with both. So we'll go through our answer choices. NT is wild type for this. I'll also just make a note when NT equals wild type. I think it's normal type is how this typed out. So NT versus MB knockout mice, that sounds good to me so far, but both treated with PX7, we want if ZEP-induced increases in PX7 requires MB, not if PX7 is there or not. So that doesn't address our ZEP-induced increase part. B, NT versus MB knockout mice, both treated with ZEP. That sounds like the simplest answer, and it will help us draw a direct line between the two, so I will choose that. But I can still go through the other ones and see that B is the best one. NT versus mice treated with MB versus NT mice treated with ZEP. So this won't let us have a direct, it won't let us have a direct comparison, nor will this tell us like if MB is necessary for the ZEP-induced PX expression, because we have nothing to compare the administration to. There's no negative control. NT mice treated with MB versus NT mice treated with NPX. So NPX is bringing in another variable that we do not want. Okay, question 5. If PX7 enhances membrane localization of ZNR5, which we know is a transmembrane receptor, and that's a GPCR, which of the cellular processes would most likely increase? Okay, so increase in ZNR5. It is also very helpful to rephrase the question, because many times when we're looking at the answer choices, we're forgetting what the question even asked us. So what did the question ask us? Increase in ZNR5 will lead to what? Knowing that it's a GPCR, we also answered it in question 1. So we want to answer whatever has the properties of GPCR, which is G protein, GTP proteins, they're hydrolyzed, and GDP is there. Then the alpha subchain detaches, all that good stuff. Or alpha subunit, my bad. Phosphorylation of tyrosine residues, not correct. Hydrolysis of GTP by G proteins. In our breakdown, we literally said that. That's the best answer. Usually, I wouldn't even read the other ones because unless there's something that I still don't understand. Depolarization via voltage-gated sodium channels. It's a ligand-gated channel. Passive diffusion of ions. Again, not right. B is correct. We'll choose that and move on. If researchers doubled the NPX dose, look at our breakdown. NPX is inhibitory. So whatever whenever more NPX is administered, we're expecting lower levels of MB and therefore lower levels of PX7, right? So we'll also just make a note of it here. NPX leads to lower MB and lower PX7. Used in figure 3, so figure 3 said NPX interferes with Zep's ability to increase MB. So what will most likely occur to MB levels relative to the original NPX condition? MB will be lowered even more, so C is correct. Choose that and move on. When you try to reason if the other ones are correct, you will lose what the question is asking. Based on our breakdown, MB levels are decreased, NPX is inhibitory, NB levels would decrease even further. C is the only correct answer. Question 7, in figure 2, why is PX7 only group expected to have CMP levels similar to control? Okay. Figure 2, PX7 enhances ZNR5 signaling, right? So PX7's role is to help ZNR5 localize to the membrane, right? Without ZNR5, ZNT cannot activate signaling because PX7 leads to ZNR5 to go all the way up to the membrane and localize there, and then ZNT can interact. So this is what's happening in my head. Like, this is how PX7 is working, right? So the one that tells us this is PX7 does not bind ZNT directly. Because PX7 blocks CMP synthesis when alone, that would be lower than the control. So we don't want that. PX7 prevents ZNR5 degradation only when ZNT is absent. None of our figures told us this. And then PX7 directly activates adenyl cyclase. No, figure 1 and 2 told us that PX7's role is that it directly interacts with ZNR5 and it helps it localize. So this is just a brief breakdown of each figures and a flowchart that helped us answer each of these questions. I also hope writing out the independent and dependent variable helps. Sometimes it can be too much to do, but with passages like this, it's easy to answer each question, even if you don't know much about the topic. As you can see, out of the 7 questions, only 2 of them needed to use background and content knowledge, and most of them were answered pretty easily using our passage breakdown. If you have any other questions, please feel free to email me.